Today I would like to say something about the possibilities of using of microbial cells in valorization of anthropogenic waste. First, see the question. Oh, I can't. No. Okay. The second one. The question is, what is really anthropogenic waste? Anthropogenic waste, it is every every type of waste generated during our life. For example, organic from our kitchen, restaurant, markets, electronic waste. In this category, we have all electronic trashes and special kind of waste, medical waste. This kind of waste is very hard to management because in this type we have plastic, glasses, biological active compounds, some pathogens, and that is why it's very hard to destroy this waste in the safe way. But the last part of the waste, and this part I would like to present more detailedly, are textiles. Unfortunately, this type of waste, textiles, isn't categorized as their own kind of waste, but together with plastics, glasses, and others. That is why it is very hard to remove this waste from our environment, and it is very hard to choose a right way to manage this waste. Every year there are produced a very huge amount of new clothes and other textiles, and a lot of them entered, as you can see, as landfills, especially in Atacama Desert in northern Chile. This is most famous landfill on the world. The second place famous for this human activity is Kenya. It is estimated that up to 60,000 tons of unneeded clothing are sent on these deserts every year. And we must know that this portion of the waste stay in this place for 200 years. And the problem is bigger because during the dumping of these textiles, about 4% of global greenhouse gas is transferred to our atmosphere, and additionally, the textile industry must consume a huge amount of water, and that is why the simple and cheap methods of recycling or reusing of this material is very important. What we can say what's more about the textile waste. Over the 70% of this type of waste are incinerated or dumped, but only 20% can be recycled at the moment. Less than 0.5% is recycled into new clothes, because a lot of clothes which ended on the landfills are still in the good conditions and can be used once more. That is why we should plan our future acting in this area according to the rules of TRs. reduce of new clothing production, recycled and reusing. And we must realize that over the 90 million of metric tons of waste textiles are generated annually. And it's a huge amount of this type of waste. And it is a big economical problem because of lacking of appropriate management of this type of waste, because of lacking of recycling, of reusing, there is lost over the $5,000 billion every year. And what's more, we planned to produce more and more fibers, and the production is still in increase. We must remember that the textile waste are composed from two main parts: wastewater and fibers. Wastewaters can be contaminated with different chemicals used for production of fibers and in every stage of processing. And as wastewater can be released to the soil, water, rivers, and can be transferred to our body. For example, as you can see on the slide, production of one shirt takes a huge amount of water. This amount of water is enough. It's enough to be drinking water, to be used drinking water for one person for 2.5 years. The second part of textile waste are fibers. The fibers can be built in different ways. Very often the textiles are composed from different kind of fibers. That is why the destruction of this waste is very hard. And the problem is with appropriate sorting of this material, because we all have tendency to cut the labels of our clothes, and then we don't know from how kind of fibers this trash is composed. Of course, we can recycle and reusing this type of waste. A lot of clothes are still in good condition, as I just mentioned, and can be transformed into another type of clothes in both open and closed loop. But the waste, textile waste, can be transformed in another products which can be used by in various industries to synthesis of novel products. And the different kind of fibers which we can find in textiles. The natural one, as cellulose, for example, of plant origin, and in the type of animal-derived fibers, like silk or wool, we can find fibrin, protein. We used some artificial fibers, which is built from natural polymers but modified by man, as in the case of viscose. And the last part of fibers, the fully synthetic fibers, as a PAT, which is made from monomers synthesized by man, or from beginning to the end. Now, the most processes used for which can be used for the recycling of this type of waste are based on the chemical or physical processes, but biotechnology can offer two ways to valorization of this type of waste. Biotechnological fermentation, in which we cultivate the microorganisms in defined conditions and use the waste as a source of carbon and energy, and microorganisms under controlled conditions can produce valuable products, for example, organic acids, bioethanol, or biogas. And we can use another approach as a biotransformation. In this type of process, we use the textile waste as a substrate for production of different products by using biocatalyst, for example, enzymes, or isolated enzymes, or whole cells of microorganisms. And as I just said, the composition of the material of the textiles is crucial for choosing the right process of degradation or revalorization, because the textiles can be built from, for example, wool, cotton, and polyester. And if we want to recover of polyester fraction, firstly, we must remove the natural part by, for example, enzymatic degradation. And we must know which enzyme is appropriate for cotton degradation and which is good for the wool degradation. And we know that for cotton, which is built from cellulose, the cellulase is the first choice. For proteolytic activity, the keratinases may be used as a degradative factor. And we know some microorganisms which are able to produce a lot of cellulases with good activity, for example, fungal strains, Trichoderma, Chaetonium, and Aspergillus. And for keratinase production, we can use, for example, Alternaria, Microsporum, Bacillus, Pseudomonas, and Streptomyces. And we must remember that this kind of enzymes are produced extracellular. That is why it is simple to induct this production and obtain the enzyme cellulases or keratinases in huge amount. And some words about the enzymes with cellulosic activity produced by fungal cells. In fungal cells, the cellulosic enzymes are produced as a complex of proteins composed of the 3 or 4 different enzymes with different specificity to place where they hydrolyze the glycosidic bonds between the glucose in the cellulosic chain. And now, for example, endoglucanase cut the chain inside the cellulose. Exoglucanase cut the cellulose and can release the simply glucose from the reducing end of the chain of cellulose. And beta glycosidase can hydrolyze glycosidic bonds between glucose in cellulose and can release the glucose from non-reducing end. And after degradation of, after acting of these all enzymes, as a result, we can obtain the solution of simple sugars, which can be used, for example, in a fermentation process for production of bioethanol, or can be used by microbes as a source of energy and carbon simply to grow. But we must remember that the textiles very often have different contamination, different chemicals. For example, dyes, which can act as an inhibitor of these enzymes and can act as a barrier and avoid the enzymes to access to hydrolytic place. That is why the textiles before the right degradation process must be pre-treated to remove all impurities, to remove dyes, and the enzymatic degradation can be realized on the clean textile. And our idea of our research is based on the selection of microbes which is able to produce cellulose, of course, and which will be able to degrade cellulosic part in the textiles directly in the sample of textiles without primary separation of the natural part from a synthetic one. And after the degradation process, the released sugars can be used for microbes growing if we use the whole cells of microorganisms, or can be used for another microorganism as a source of carbon for fermentation process. And the synthetic fibers, which we hope will be untouched, can be derived to the recycling. And as the first, we selected two microorganisms, Trichoderma viride, which is known of cellulitic enzymes producers, and Ideonella saccharensis, bacterial strain, which is famous for another activities. This bacteria is very often called as plastic-eating bacteria because it's able to fully degrade PAT. First step of degradation is hydrolysis to monomers of PAT by PATase activity, and then these obtained monomers are degraded to simpler products, ethylene glycol and terephthalic acid. And in the natural conditions, these compounds can be used for these bacteria on another bacteria in consortia to mineralize the plastics. And this is the main function of this bacteria. But we want to use these microbes to removing of cellulose, and we hope that it will be possible to stop the reaction for the first enzymatic activities, and obtained monomers of PAT can be used for synthesis of another polymers. And our preliminary results, we at the first stage we decided to induce cellulase production in the case of trichoderma. In the case of trichoderma, we use as the first stage culture PDB medium, two kinds of this media, commercially available, supplemented with glucose, and another one prepared in home, contaminated with carboxymethylcellulose. Carboxymethylcellulose was our model substrate, and in theory, this modified cellulose is better soluble in water. In theory, in practice, it's very hard to make a solution of this polymer. It is needed to use very small concentration and very high temperature and very long mixing. But after obtaining cells, the biomass was transferred to another induction media, and at the second stage, the media was more defined as a potato dextrose medium, we used mineral medium and Mendel and Andreotti medium supplemented with carboxymethyl cellulose, or at the first time glucose. But the other experiments were done only with carboxymethyl and cellulose and crystallic cellulose. And after the appropriate time, we obtained some samples of post-culture media, and which were tested for cellulitic activity by simply test with using of water agar supplemented with carboxymethyl after appropriate time. All plates were floated with Lugol's liquid, and we can observe the clear zones in some cases. And as you can see on the picture, we selected the 4 different conditions in which the cellulase activity was higher, and for these purposes, the appropriate was mineral medium supplemented with carboxymethyl or cellulose, and the cultivation induction time take place 3 or 6 days. And it was in the case of Trichoderma viridie, it is the simpler microorganism to induce activity. In the case of Ideonella saccharensis, the situation was more difficult because this microbe is famous from degradative activity to synthetic polymers, but cellulitic activity was rather was rather low after the basic stimulation by cultivation on the medium supplemented just by carboxymethyl. And in this case, we must use the special multi-step stimulation when 3 times we cultivate the Ideonella on the medium supplemented with carboxymethyl, and the last step is the cultivation with using of cellulose as the model substrate. And after these operations, we obtain much higher activity of cellulases. This plate, which was prepared in the same way as the others, was made by using of commercially available cellulose solution derived from Aspergillus niger. This plate is to comparison of the level of activity in the defined solution of cellulase and in our microbes. And the second part of the determination of cellulitic activity was to measurement of the released simple sugars, simple reductive sugars by using of DNS method. This method is based on the using of dinitrosalicylic acid reagent. And after the measurement of absorbance, we calculate the amount of glucose by using of standard curve, which was prepared earlier, of course. And after all of this, we selected some conditions, and we prepared the main experiments in which we used the sample of textiles built from cotton, PAT, and elastan, and we used small pieces of this material, which were crushed, washed, and stirred with sodium base. This stage is very important because the cellulose is resistant to degradation because of hydrogen bonding between the chains of cellulose, and this part of experiment should partly destroy these hydrogen interactions. And after shaking, the fibers were washed, dried, and transferred to the enzymes cocktail obtained in the induction step. After 7 days of degradation of recycling degradation process, we isolated the fibers, washed carefully with water and ethanol, and prepared for microscopic analysis. And of course, during the whole experiment, we take every day a small amount of post-reaction media and tested for reducing sugars by using this method. And unfortunately, we now still are waiting for the microscopic analysis because we produced a lot of samples and it takes time to prepare these samples to analysis and analysis themselves. But based it on the mass loss and the amount of release of glucose, we can say that some conditions most promising, and for example, for Trichoderma viride, cultivation of mineral medium supplemented with carboxymethylcellulose or cellulose by 3 days, and the pretreatment of the textile sample in sodium base for 4 hours. In the case of iodinylase analysis, of course, we must use the additional stimulation with carboxymethylcellulose and cellulose, and we must treat the textile samples with the using of sodium base for 1 or 4 hours. And we now know that the pretreatment is very important because, as you can see on the control pictures, the fibers built from cotton and PAT are very resistant to degradation. These fibers have the very regular shape, and to facilitate the enzyme to attach and degrade some of the cellulose, we must release a little structure of cellulotic part. And the second stage, very important, is the right washing after the degradation process, because the proteins, the cellulases, attached on the surface of the fibers, and before prepare the samples for microscopic analysis, we must remove all of these proteins. And as I just said, there are very, very preliminary results. We hope that we find the appropriate conditions for degradation of the cellulosic part, and the synthetic part stays untouched. But for proper degradation process, or the proper recycling process, it is very important knowledge about the composition of the material. That is why my personal ask for you, don't cut the labels. And thank you very much for your attention.